Hi everyone, my name is Colleen Carpenter and I'm currently an assistant professor at the University of Richmond. I started here in January. I am honored to be a part of this lineup of amazing scientists today, though I am sad I can't join this session in person. As we're all aware, LGS is a severe childhood epilepsy and there is urgent need for new and effective therapeutics for this disease. Today, I want to present the results of a large-scale project that used zebrafish to functionally evaluate the genetic drivers behind catastrophic pediatric epilepsies, including LGS. This work was done during my time in the Bioben lab at UCSF and involved many, I can't stress many enough, talented hands, which I will highlight at the end of this talk. I have no disclosures. So why zebrafish? What's so cool about this organism? I will tell you about that in this slide. Humans and zebrafish share approximately 70% of the same genes. When we think of disease-causing genes, that number goes above the 80s. Zebrafish adults, so an adult female zebrafish can generate up to 20 or 250 eggs per mating event. So if you think of answering your basic science questions and having a large enough sample size, zebrafish provide that per mating event. And you can breed zebrafish multiple times per week. Let me not oversell it. Let's say twice per week. The embryos are fertilized externally, so you can access, you can ask early stage developmental questions in a pretty complex organism. These embryos are transparent, early stage larvae are transparent, so you could do really nice imaging studies looking at the whole brain with understanding circuitry and signaling. And in thinking of a brain disorder like epilepsy, you can imagine the advantages of that. I'm a pharmacologist by training, so I'm always thinking, okay, how do we improve drug discovery? How do we do these experiments in an effective way? And there's an ease with using zebrafish for drug discovery experiments. So drugs can be directly administered to the media that they're in. So here we're showing two larvae. So these larvae are about the length of an eyelash. So, they fit nicely in the walls of a 96-well plate. And here we're testing, 200 larvae at the same time. So easy to administer the drugs, and you could test a lot. So we have that high throughputness with using these organisms. Creating transgenic models in zebrafish versus many traditional models is a lot quicker. And with thinking of the drug discovery pipeline and thinking of inefficiencies, I think there are many points where we can add new tools that can make it cost effective. Right now, it costs about $3 billion to get a drug to market. So just thinking of, the low cost and high throughputness of zebrafish, I think that makes it a very attractive model for drug discovery. So they're pretty cool. But is modeling epilepsy in zebrafish actually feasible? Yes, and I start with a yes and a lot of optimism based on the incredible success of the Dravet syndrome zebrafish model. So patients with Dravet syndrome tend to have loss of function mutations in SCN1A, which encodes for an NAV1.1, a voltage-gated sodium channel. So SCN1LAB is a zebrafish homologue of SCN1A. And SCN1LAB mutants, so knockout larvae, exhibit electrographic seizures, as shown in this diagram on the left. So this is looking at wild type or control larvae. So, normally we don't see much electrical activity under, normal circumstances. But when we knock out this gene in zebrafish, at early stages, so this is DPF is days post-fertilization, we see these large amplitude, long duration electrical events that are very reminiscent of electrical seizures that we see in patients. And these fish also exhibit behavioral or seizure-like behaviors. And here I think they look a lot like tonic-clonic seizures. So I'm going to show you what the fish normally do. So this is a 96 well plate. We have our larvae. It's habituated, so it knows there's, you know, it's a new environment compared to probably the petri dish it's been living in. But over time, it knows that, okay, this is a cool setup. It's not doing much. This is a regular swim behavior. For somebody who does a lot of behavioral recording, you know, this gets pretty boring really quickly. But when we look at our mutant fish, what we see are these spontaneous high velocity convulsive light movements. And what I mean by that is, you know, we're seeing almost this like thrashing across the well, and we see this quite frequently in this line. So I want to stress spontaneous here, because with thinking of epilepsy, it's characterized by recurrent spontaneous seizures. So in creating valid models of epilepsy and using zebrafish, we wanted to see spontaneous events. So these fish are producing the spontaneous electrical events and the spontaneous behavioral events. We also investigated traits associated, other comorbidities associated with Dravet syndrome, such as sleeping disturbances, and these fish effectively recapitulated many of those traits. So definitely, you know, and at this point, we know a valid model of Dravet syndrome. But one of the huge advantages, again, of using zebrafish is, you know, if we now have a model, it has a potential for drug screening efforts. So with the Dravet line, and this is like speeding up the story, right? These fish were used to screen over 3,500 drugs, and now successful drugs that all seem to modulate serotonin receptors have gotten orphan drug designations, and clemizole, which is highlighted here. So these are just showing them blocking the electrical seizures, are now in phase 2 clinical trials. So huge success, right? And when we think of the traditional way of doing things, this is skipping a lot of steps. Here we're moving from zebrafish to people. So it gives us hope in, you know, tweaking and improving the normal pipeline for getting drugs to the people that need them, and with tweaking it in a way that I think will be more efficient and more cost effective. So very, very exciting, right? So with this excitement, we translated this to the easy project. It's like, okay, I can do it with one pediatric epilepsy. Let's do it with all the others, which is a huge goal, and a lot of work. But I am proud to show the results of this study, because I think it further supports the use of zebrafish as a model, to be used as a model for epilepsy. So in the Epilepsy Zebrafish Project, EZP, we use fish to understand the causative role of de novo single gene mutations in the development of other pediatric epilepsies, including LGS. So using GWAS studies, coupled with homology and gene expression studies, we created 40 zebrafish lines that mimic loss of function mutations found in epileptic patients. We then phenotyped these fish using morphological, survival, electrophysiological, and behavioral assays, looking for traits of epilepsy. Zebrafish were embedded in agarose, and local field potential readings were taken from the tectum, and type 0 to 2 events were noted. So type 0 events are basal activity, type 1 events are low amplitude interictal-like sharp waveforms, and type 2 are really our seizure behaviors or seizure-like events, which are large amplitude, ictal-like, multi-wave or multi-spike waveforms. So we're looking for fish having recurrent type 2 events. So this figure summarizes our electrophysiology findings. Bolded genes represent models with significant ictal activity, which we classify as our epileptic lines. So we're not saying here that other genes studied have no contributions to epilepsy, but just that only roughly 20% of these zebrafish lines are robust epilepsy lines that support the causative role of single gene mutations in the development of various pediatric epilepsies. I've highlighted genes related to LGS, and we believe that our GABA beta 3 and SCXBP1 models effectively show facets of LGS. one thing I quickly want to address is there's always questions, or we always get questions of, do wild types exhibit type 2 events? We do see them rarely in our wild types, but comparing them to our epileptic lines, we see that the epileptic lines have significantly more type 2 events when we compare them to the other events that we monitor. Since epilepsy tends to increase the risk of premature death, we investigated early fatality in our zebrafish lines and saw this most significantly in the highlighted lines. So other zebrafish models of ALDH7A1 and DEPDC5 deficiencies also confirm these data. So no, this is a shortened survival assay looking at fatalities before 14 DPF. So I do think if we extend the study, we would have likely seen other fatality traits in some of the other lines that we created. Through these studies, we also refined our classification of behavioral seizures. So using DanioVision video tracking and in-house algorithms, we analyzed PTZ, so chemically induced seizures, and spontaneous behavioral seizures in SCN1 LAB mutant zebrafish to create a threshold for classifying behavioral seizures. And these are more tonic-clonic-like seizures. So here I'm showing that, what we see is wild-type fish will have low speed to high speed events. But what we see is that epileptic lines have these long duration, high speed events that the cutoff is events that are greater than 28 millimeters per second, and they're about equal to or greater than 1 second in duration. So this is on the time scale actually of the electrical seizures that we normally see in zebrafish. So this is what we classify now as our seizure-like movements. So validating our assays, so this is looking at PTZ-induced seizures. So with increasing concentrations of PTZ, using the algorithm, we see an increase in seizure-like events, which is what we expect. It's just validating our method to classify behavioral seizures. So this is just a snippet of the behavioral data we got from the study. So at the end of it, what we actually found is the only line that showed the behavioral seizures were the Dravet line, which we had seen before. All our other epileptic lines actually don't show these behavioral seizures. But then the question is, we're characterizing one type of behavioral seizures. So this kind of opens a space for us to figure out how to use behavioral tracking to mimic other types of behavioral seizures. I will point out that our ARIX-A mutant line exhibited hypoactivity, and this is a trait I've been seeing in a couple epileptic models, and I think it's interesting, or it will be interesting for us to further understand what this hypoactivity is really reflecting. So with thinking of LGS, you think of the immediate seizures, but then you have to think about the comorbidities, because it's the comorbidities that also affect, everything together affects the quality of life, right? You could decrease seizures with some treatments on the market, but we have patients that have some alleviation of seizures, but they still have significant problems with the comorbidities that they're dealing with. So with creating a model and looking at LGS zebrafish models, we want to create models that show the electrical seizures, potentially show the behavioral seizures, but also the comorbidities as well. So when we're doing our drug screening, we can look for candidates that affect all the different traits that we see in our patients. So with looking at our current models of LGS, we've started to look at them in different assays. So what I've been doing, and this is unpublished work, is starting to look at some of the exploratory behaviors, looking at development and neurodevelopment in these lines. And I just wanted to present a couple assays that I've done so far. So investigate exploratory behaviors in zebrafish, and also this kind of touches on anxiety-like behaviors. I've been testing different lines in a light-dark preference assay. So larvae were placed, so it's a tweaked light-dark assay. So larvae were placed at the boundary of equally sized dark and light arenas, and then we tracked the distance traveled and time spent in these different arenas. So normally, larval zebrafish display an innate preference for the light, or they exhibit a light preference. However, when given the choice, we found that SCN1 LAB, so Dravet mutant zebrafish, show a dark preference and reduced exploratory behaviors. And we think this is also a reflection of deficits in development in this line. So I've been using this assay to also explore deficits in STXBP1B, the STXBP1B mutant zebrafish. And we're also seeing trends that are similar to what we've seen with our Dravet line, where they do seem to have a preference for the dark arena, and that they, when there, they do explore it more. So the question is, what is this dark preference really a reflection of when we think of symptoms or traits that we see in patients? And I think, you know, there's a lot more work to be done to really understand what these deficits really mean that we're looking at in the fish. So one other thing that I began to look at is learning deficits and motor dysfunction. So normal startle responses in fish are observed as a pattern of rapid contractions of musculature that affect locomotor activity. So deviations from the normal response pattern will give insight into motor function and deficits in basic forms of learning. So to assess unconditioned startle responses, zebrafish larvae were habituated in a 96 well plate in the dark, and then they were subjected to a sequence of light flashes. So as you can see, the mutants, so the STXBP1B mutants, had a significantly impaired response, providing some preliminary evidence that this model can recapitulate motor and learning deficits associated with LGS. So now we have a rich resource of models for further understanding and hopefully treating LGS and other epilepsies. You know, I didn't have too much time, but I will put a little plug for some work done by one of my other lab mates while I was at UCSF, Jing Lu. So she did some beautiful work looking at calcium signaling in STXBP1B mutants, which show the presence of network cascades, and I think lays a foundation for the validation of functional biomarkers of the epileptic brain. So there's so much potential here. I think this is a great way to really address many of the unanswered basic science questions we do have about LGS and other epilepsies. And I hope I showed you, you know, zebrafish provides a platform for that, but also we hope a platform for personalized medicine. I'd like to end by thanking all the people on this slide, especially Scott for letting me join the lab. I joined the lab as a pharmacologist. I didn't know about epilepsy because I was in the pharmacology department at University of Michigan, and you know, between Lori's lab and everybody else that does epilepsy in Michigan, I did learn a lot. So I knew a lot on the basic science side, but I definitely didn't know how to work with zebrafish. So that I learned completely in the Baraband lab, and I'm so thankful for that experience. And obviously was inspired by that experience because that's some of the work I'm doing in my lab right now, particularly focused on understanding the development of LGS. So thank you, Scott. Thanks to everyone on this slide. Thanks to the LGS Foundation for supporting me. Really, really appreciate it. I hope everyone enjoys the rest of the session. Thank you. Bye.